Okay, so we'll start talking about genetics. There's a few terms that we need to get straight. The first one is allele. So we may know alleles as either uppercase letter or lowercase letter. So let's say we have big B, little b. These are types of alleles. So let's say the big B stands for black fur. This is the dominant one. And let's say the little b, if you get little b, is for white fur. So if we throw in some of these vocabulary words that you guys have, we would call this the dominant trait. Usually the capital letter represents the dominant, and the lowercase letter usually represents the recessive. So what does dominant and what does recessive actually mean? So if we go over those terms, dominant would mean as long as you have one of those letters, so as long as you have a big B, you're going to have that dominant trait. Recessive is a hidden trait. So you would have to have two little b's in order to have that particular trait. So as an example, if we have big B, big B, big B, and little b. These are kind of the three different combinations of how we could put these letters together. So an allele is just a type of a trait. So either you can have the black allele, or you can have the white allele, or you can have one of each. So for this one, we call this homozygous dominant. Homo meaning the same. So homozygous dominant meaning they're the same letter or same allele, and they happen to both be the dominant allele. So homozygous dominant. This one we refer to as heterozygous. Hetero meaning different. So there's one of each. There's a big B and a little b. And this one we refer to as homozygous. So again, homo meaning the same. This is homozygous recessive. So they're the same and they both happen to be the recessive one. So if you're ever given a Punnett square where they say the heterozygous, which would be this one, versus or crossed with the homozygous dominant, that's this one. So they may use these terms and they would expect you to know what kind of letter combinations or what allele combinations are expected from them. So let's also throw in some other terms that you guys may have used. So all of these can also be called genotypes. A genotype is your allele combination or the letter combinations. So if someone is big B, or if someone is homozygous dominant, that's their genotype. Someone else's genotype could be heterozygous. So genotype is always your allele combinations or the letter combinations. You guys also have the word phenotype. So let's talk about what phenotype would be. So in our example, big B is for black, little b is for white. So if you are big B, you would actually have black fur. Okay. So you can see that your genotype actually can tell us what you physically look like. So you'd have black fur. If you are big B, little b, you also will have black fur. So one thing to keep in mind, and you guys may have used the term Mendelian genetics, that just means Gregor Mendel was the scientist that studied genetics. And so according to his theories, as long as you have one big B or one dominant allele, you automatically have that trait. So even though this one has a big B and a little b, it still has that same black color fur because it has that big B. Okay. This one that has two little b's, so it does not have a big B, this one would have white fur. Okay. And again, it doesn't have the black fur because black you have to have at least one big B to have black. This one has two little b's, so it's going to have the white color. Now, the different color types that we could actually see with our own eyes, so the black, and the white here, these guys are phenotypes. So phenotypes are your physical characteristics. So what do you physically look like? So someone may have brown eyes or green eyes or blue eyes or black fur or white fur. So those would be phenotypes, things that we can physically see with our eyes. The genotypes, these guys here, that's something that we can't necessarily see by looking at someone. We'd have to kind of draw their blood and do a karyotype or take a picture of their chromosomes to see what type of letter combinations they have. Okay. So let's do another example. So let's say we have a organism that has, if we're doing our same white fur, black fur kind of scenario, let's say we had a rabbit. Let's talk about rabbits. Let's say we have a rabbit and our rabbit has black fur. So let's say we see physically their phenotype. Their phenotype is black fur. But we want to know their genotype. Well, one of the issues with black fur is that we saw in our other example, there were two different ways that you could have black fur. You could have a big B could give you black fur, but big B, little b could also give you black fur. So by looking at a rabbit or an animal that would have, let's say, black fur as an example, it'd be hard to figure out which genotype they have. Would that be the same if they had white fur? So let's see if we could see a rabbit that had white fur. So the phenotype would be white fur. Well, the good thing about this is what we call the recessive phenotype, meaning you can only have this color fur if you have two of these. So for the recessive, if we see white fur, we know automatically that their genotype would have to be little b. Just because that's the only way that we can see recessive traits is if we have two of the same things. So recessive traits, like in our case white fur, are really good because not only can we see them, but we know exactly what their genotype are. Dominant traits, like with the black, it's hard to say. Now we can see what color they are, but then it would be hard to figure out which one of these two it would be. Okay? So that introduces something called a test cross. So Gregor Mendel, the scientist that studied genetics, he took a bunch of pea plants and he crossed them with different combinations just to see what types of new combinations he can make. And based off of the crosses that he did, or the mating patterns that he did, he was able to work backwards and find genotypes. So we're going to do something called a test cross. So a test cross is helpful if you are trying to figure out the genotypes. If you're trying to figure out the letters, it's helpful if you're trying to figure out the letters or the genotype of an organism. So let's say we have a rabbit that has black fur, and we saw with black fur, it's hard to figure out whether it's big B, or big B, little b. So let's say we have a rabbit, we know that it has black fur, we can physically see that. But we want to figure out for sure whether it's big B, or big B, little b. We can cross it with something that we do know. Well, remember we said with white fur, we know exactly what your genotype is. So we're going to cross that, and the X just means crossing or mating them with, let's say, a white furred rabbit. Okay. And what we'd want to look at for this test cross to be able to work backwards and figure out which one of these this guy had to have been, we can look at the combinations of their offspring, how their offspring appear. So as a hypothetical, let's hypothetically say that the black fur was big B, big B. Okay, we know that the white fur is little b. So for our Punnett squares, we draw our square and we separate these guys. So big B, and we'll separate these two, little b. So one thing that we noticed that hypothetically, if this black fur was homozygous dominant, meaning they had two of the big B's, all of their offspring should all have black fur. They all should be heterozygous, but we said that heterozygous still also has black. So if this rabbit were big B, all of their offspring should be black. There's no way that their offspring could ever be white because none of these combinations are little b. Okay? All right, so let's look at the other scenario. Hypothetically speaking, let's actually assume now that that black fur was heterozygous. Let's assume that it was big B, little b. Okay? We are still crossing it with white fur. And we know white fur is little b. Okay? Now in this case, when we do our Punnett square, we'll separate this guy here. We'll separate these little b here. So now we see a little more variety. Now we actually are able to see that hypothetically, if that black rabbit were big B, little b, that we have a little more variety. We could actually have half or 50% could be black. Half of their offspring could be black, but half of their offspring could also be white. So what we would do for a test cross, if we ever were trying to figure out a genotype, is we would cross that organism that we don't know the genotype. So in our case, we didn't know the genotype of the black fur rabbit. We cross it with one that we do know the genotype. We always cross it with one that's recessive, and we would look at the offspring. So if all the offspring came out black, then that means that that black furred rabbit had to be big B. If when we cross them, all of their offspring come out black and some also come out white, then that means that that black fur had to be big B, little b. So anytime you have a test cross, you just cross that unknown with a homozygous recessive, and you look at what the offspring are. So that way you could work backwards and figure out what the parent must have been, what the unknown parent must have been. So while we're on that, let's talk about some of the definitions that you may have to know for this. There are a couple of Mendel's laws that you have to know. So let me just erase this part up here. So Punnett squares are usually pretty straightforward, but there are some terms or some assumptions that we assume are true, which is why we're able to do some of these Punnett squares. And these are the three of Mendel's laws. The first one we'll talk about is the law of segregation. So Mendel's law of segregation. Okay. Segregation means to separate or keep things separated. So when we do our Punnett squares, so let's say for example we have big B, little b, and big B, big B. When we write our Punnett squares, we actually already do his law. We actually separate these guys. So we say that we put one big B here, a little b here, and we separate these two. And so what actually happens when we do our Punnett squares is that each parent is only giving one allele. So this parent could either give a big B to this child, or this parent could give a little b. So law of segregation is just the idea that alleles, or our letters, alleles separate. Okay. So in a parent, even though a parent may be big B, little b, it only can pass on one of those alleles. So those letters have to separate. Mendel's second law is the law of independent assortment. Independent just means something is not relying on something else. Assortment just means variety. So law of independent assortment just means that even though these alleles can separate, nothing says that this big B has to always go to all their offspring, or that this little b has to always go to all their offspring. It's a mix and match as to which one of these combinations the child will get. So not only can these alleles separate, but they can recombine in different orders. Okay. And then the third one that we have is the law of dominance. And we'll go through a couple examples of the law of dominance can have some exceptions, like incomplete dominance and codominance. Those are under this law of dominance. There are a little bit of exceptions though. But the law of dominance just says that as long as you have at least one dominant allele, you have the dominant trait. So at least one dominant allele means you have the dominant trait. Now what does that actually mean? So we said, like for our black fur example, big B is black. Big B, little b is also black. So according to Mendel's law of dominance, as long as you have at least one capital letter or at least one dominant allele, you have that dominant trait. So this guy, even though it only has one big B, the fact that it has at least one big B means it has that dominant trait. It still is black. Now this changes when we deal with different types of dominance. So let's go into those now. We can have something called codominance and we can have incomplete dominance, but those are exceptions to Mendel's law of dominance. Okay, because Mendel's law of dominance just says you need at least one big letter, one dominant allele to have the dominant trait. So these are exceptions to the law of dominance. The first one we'll talk about is incomplete. Incomplete dominance. Okay. So incomplete dominance, we go through an example. Let's say if you have big R, you have a big R allele, then you will be red in color. Let's say maybe this is colors of a flower petal or something. If you have the little r, let's say you'd be white. You'd have the white petals, okay? So normally we would think big R would be red in color. Little r would be white. So those are easy to predict. But with incomplete dominance, the heterozygous, so the one that has big R and the one that has little r, is not going to be red, because we would predict it to be red according to Mendel's law of dominance. But with incomplete dominance, the one that has one of each is actually a blend of the two colors, so it would be pink. So incomplete dominance, that's when your heterozygous, or the one that has a big letter and a little letter, is a blend. Okay? So red and white would give us pink. If we did our black and white, for example, this would be black, that would be white, but the big B, little b would be gray, or somewhere in between. So incomplete, only the heterozygous is a blend of the two colors, or of the two traits. So let's do another example for incomplete. Let's say big T, if you had big T, you would be a tall plant. Let's say little t, you'd be a short plant. So big T, we know would be tall. We know little t would be short. What about big T, little t, if you have one of each? Well, according to incomplete dominance, you actually would be medium height, somewhere in between the two. So if this one's tall and that one's short, you'd be right smack in the middle. Well, now for a word problem, they would have to tell you that it's incomplete dominance. Otherwise, you would have to assume that the heterozygous would be tall. So unless they tell you that it's incomplete dominance, we have to assume that as long as you have a big T, you would be tall. Okay? The other exception to Mendel's law or to the law of dominance. So one exception we said is incomplete, where you're dealing with a blend. The other exception is codominance. Now with codominance, both of the traits are present at the same time. So if we have an example, usually we see this with blood types, but I'll do it with a different example. So let's say, again, big R is red. Let's say little r is white. Okay. So we know big R would be a red colored plant. We know little r would be white. Okay, now we know with incomplete dominance, the big R, little r would be pink. If it were incomplete, we blend them together. But codominance is not about blending. In codominance, you'd actually see both of them at the same time. So a big R, little r would be red and white. Maybe red with white polka dots or white with red polka dots or something like that. But you would see both of the colors together. So codominance is both traits are visible. Okay? And this only again applies to the heterozygous. Because it's straightforward when we have big R, or little r, but for the heterozygous, that's where it gets a little bit iffy. So just to recap, if we're dealing with the normal dominance, which is Mendel's law of dominance, the heterozygous should look exactly like the dominant. So if the dominant is black, the heterozygous should look black. For his exceptions, if you're dealing with incomplete dominance, your heterozygous is a blend of the two colors or a blend of the two traits. So if big R is red, or if big T is tall, and little r is white, and little t is short, the heterozygous would be right in between. So if we have red and white, the heterozygous would look pink. If we have tall and short, the heterozygous would look medium, somewhere in between. Codominance, again, is another exception to Mendel's rules. If we have big R is red, little r is white, the big and little together, so the heterozygous, you'd see red and white together. You'd see both of them. So incomplete is always about blending the two together. Codominance, if you think of cooperate, they're both there. So you see both of them together. Only again for the heterozygous. So unless they tell you in the question, we have to assume that we're dealing with the normal type here, where the heterozygous looks like the homozygous dominant, unless they tell you otherwise. A couple other terms that you guys will definitely need to know for the quiz is multiple alleles and polygenic traits. So I'll see if I can come up with a couple of examples. So for multiple alleles, the only example we really have that you guys may have talked about is blood types or blood groups. And so normally when we're dealing with these guys, when we deal with alleles, so if we're talking about multiple alleles, again, alleles are kind of choices or options. That's like the big B, the little b, the letters that we talk about. Normally we only have two options. Normally we only have a big B for your dominant, or we have a capital letter for dominant, and usually we have a lowercase for recessive. That's usually the normal case. But for multiple alleles, we have multiple options. We don't just have dominant and recessive. We have a little more to choose from. So the example with blood group, I'll go through the example with blood group. With blood group, you have three different choices that you can inherit from your parents. You can either inherit the A trait, and for whatever reason, for blood type, they always use the letter I with a little letter up here as a superscript. So either you can inherit an A from your parent, you can inherit a B, or you can inherit an I. So it's not as just a big B and a little b. You don't just have two allele options that you can inherit. You actually have three. So this would be an example of multiple alleles. You have more than two. You have three different options. Now the good thing about having multiple alleles is then you can have multiple combinations of traits. So you can have big A, big B. You can have big A, big A. You can have big A, little I. You could have big B, big B. You could have big B, little I. You could have little I, little I. So you actually have 6 different outcomes that you could have just by adding on one extra allele. Now if we go back to our regular example, we only deal with just one letter, or the option of either having a capital letter or dominant, or a lowercase letter or recessive. So if we only have big B and little b's, we only have the option to be having big B, big B, big B, little b, or little b, little b. So we see that just with 2 alleles, you only have 3 kind of outcomes. But with 3 alleles, you actually now have 6 different outcomes. So the more alleles, or the more choices that you add for a trait, the more options that you have for your offspring. So it just creates variety. Now if we talk about polygenic traits, so multiple alleles are just kind of giving more variety. It's almost like a salad bar. You know, the difference between a salad bar with maybe only 2 options versus a salad bar with like 10 options. There's lots of different types of salads you can make, as you know, with the more salad options that you have. So if we talk about a polygenic trait, I'll fit that down here. So polygenic traits. Now we kind of simplify things when we deal with genetics in our biology classes. In reality, genetics are a lot more complicated because if you think about, everyone is slightly different. There are different skin tones, different hair colors, different hair textures. It's not as cut and dry as black and white or red and white. But polygenic traits are where there are multiple traits or multiple genes or multiple sections of the DNA that code for the same trait. Okay. So in here, you're looking for a combination of traits together that would give you a different outcome. So as an example, let's take, and this is just kind of a hypothetical, I'll make this up just to kind of explain this, but let's say we're dealing with skin color. If you think about all the different types of beiges and browns and whites that you could come up with, there's a lot of different skin tones. There's not just cut and dry as just dark brown, light brown, or white. It's a lot more of a continuum. So what actually would happen for skin color, same is true for maybe fur color for different animals, is that there's different traits or different genes, different parts of the DNA that code for that trait. So for example, for skin color, you may have to have big A, big B, big C, big D, big D. You may have to have all of these capital letters in order to have, let's say, a dark brown colored skin. But if someone were to have big A, big B, big C, big D, so everything the same, but now this is a little a, they may have a slightly lighter color skin. Whereas someone that maybe has little a, little b, little c, big D, little d, they may have an even different color skin tone. So polygenic traits are just where you have groups of alleles or groups of traits that when combined together give you that trait. So we'd have to have all 4 of these to give us our skin color. And you can imagine all the different types of combinations of letters you can get. So big A or big B, little b. And depending on the different combinations that you get will dictate your phenotype or your skin color. So polygenic traits, just to kind of summarize, they're a lot more complicated, but they're traits that require multiple genes to give you that color. And these traits are going to give you a continuum or a wider variety. Because if you can think about all the different combinations, you have all these different variations of skin color or height or weight or different things like that you can get. It's not as cut as dry. It's not as cut and dry as big T is tall and little t is short, and that's it. It's a lot more variety. The last thing I'll go over is the difference between monohybrids and dihybrids. And these are different Punnett squares or crosses that you guys may have done. So when we do the little Punnett square where it's just kind of one block, those are for monohybrid crosses. Those are usually the easier ones to work with. The dihybrid is where you really get the big block of 16, and it gives a lot more variety. So if we talk about the difference between monohybrid So monohybrid versus dihybrid. So mono means just one. We're dealing with just one trait. So maybe it is plant height or fur color or pod or pea pod shape. So let's say big W is wrinkled shape pods for a plant, and let's say little w is smooth. Well, this is where you would have something that's a lot more straightforward. So you might just have a big W crossed with a big W, little w. The idea with monohybrid is you're only dealing with one trait. And for this example, the trait will be, let's say, pod texture. We're just dealing with one trait. So it might be plant height, it might be fur color, it might be hair texture, but if you're just dealing with one trait. So you usually just have one Punnett square, where you would separate. Remember law of segregation, we can separate these alleles because the parent can only give one of these alleles at a time. And so for this option, we would see that these are the different combinations that this parent could give, these two parents could give. So this is monohybrid, we're just dealing with one trait at a time. These Punnett squares are a lot more simpler to do. Dihybrid, di means two, okay? We're dealing with two traits at a time. So we may have, let's say, big A is a rough, let's use R instead. Let's say big R is rough texture and little r is smooth. So one of the traits that we're dealing with would be, let's say, plant texture or leaf texture or something. But then they will give you a whole nother set of alleles. So let's say, so just to recap, dihybrid would be two traits. Let's say the first trait is plant texture. So big R is rough, little r is smooth. Let's say that they also give you plant height. So big T is tall, little t is short. So we're also dealing with plant height. So we have two traits that we're trying to keep track of at the same time. So this is where it gets a little more complicated. So they could give you something like big R, big T, little t is crossed with big R, little t. Okay? So we're dealing with two traits at the exact same time. Now this particular parent has the big R, so they would have the dominant, which is rough. So this would be a rough, and then big T for tall. This is a rough, tall plant. So again, we're dealing with two traits at the same time, plant texture and plant height, whereas this plant would be rough and short. Okay, short because these are both little t's, and the little t is the short phenotype. And so in this case, you may end up having a lot larger of a square. You don't always have to have a 16 or 4x4 square. It all depends on the combinations that each of these parents could give. So when we do these types of dihybrid crosses, you have to look at the combinations that the parents can give. This parent could give a big R and a big T. Remember, they can only give one of each letter. So this could give a big R and a big T, or it could give a big R and a little t, or it could give a little r and a big T. Again, I'm just looking at the combinations of how this parent could give one of each letter, or this parent could give a little r and a little t. So if we notice, this parent has 4 different combinations that they could give. Okay. What about this parent? Well, this parent only has a big R. There's 2 of them, but it's the same. So this parent could only give a big R, and it could only give a little t. There's no other variation that parent could give. So actually, in this case, we don't have one of those big Punnett squares. We just have one that's relatively short or small because this parent can only give one combination, but this parent could give 4 different combinations. So we would end up with big R, big T, little t, big R, little t. We try to group the letters together. Big R, big T, little t, and big R, little t. So these are the 4 different genotypes that their offspring could have, and we could come up with the phenotypes of what these offspring will look like. So this one would be rough, because it has the big R. This one would also be rough. This one would also be rough. And this one would also be rough. So if we're talking about just plant texture, they all would be rough. Now let's look at plant height. This one, if we're looking at just the T's, this is tall. This one's short. This one's tall. And this one's short. So if we see, there's like a 50-50 chance, 2 out of 4, that they'd be rough and short. And then there's a 50-50 chance that they'd be rough and tall. Okay? So again, I just looked at the combinations of this parent could give and the combinations of this parent could give. Okay? So let's look at a different example. So let's say we actually have 2 parents that are heterozygous. So we have one parent that's big R, big T, little t, and one parent that is big R, little r, big T, little t. Okay? So they're both heterozygous. If they're both heterozygous, then each of these parents has a bunch of different combinations that they could give. This parent could give a big R, a big T. It could give a big R, a little t. It could give a little r, a big T. It could give a little r, a little t. But this parent could give the exact same type of combinations. This parent could give a big R, big T, one of each. It could give a big R, little t. It could also give a little r, big T. And it could give a little r, little t. So if you notice, one parent has 4 combinations they could give, and this other parent has 4 combinations it also can give. This is where you would get the really big Punnett square. And so we would have a 4x4. So we have 4 different columns here. And then we would have 4 going this way. So this one is going to be a lot bigger, a lot more combinations that we could end up coming up with. So I'm not going to do all of them here in the video, but this is where you would actually have 16 different options. One other thing to keep in mind for these squares where you actually do have 16 by 16, or 4 by 4, it's only when both of the parents are heterozygous, because you have so many different options. In the example we did before, we saw that one parent only had 1 option to give, so we only just had kind of 1 column to deal with. So for dihybrid crosses, You want to always look at the combinations that each parent can give, write them on one side, look at the combinations the other parent can give, write them on the other side, and then do your square. So it does not always have to be a big 4x4 square.